Hi everyone. So in this video I will talk about post-translation in the secretory and non-secretory pathway. So let's begin with the synthesis of protein first. Um, as you know, this is the nucleus and this is the nucleolus. So what the nucleolus does, it's actually the place of the rRNA synthesis, which is the subunit of the ribosomes. So it gets out and it begins its synthesis of the ribosomes. This is for the nucleolus. For this part right here, the packed part right here, this is the DNA. So we need to transcribe that DNA to RNA. So there will be the process of transcription using RNA polymerase to synthesize mRNA. This is in the nucleus. So the mRNA would actually exit the nucleus, where it will be meeting with the ribosome right here. So this is the mRNA, and it's going inside of the ribosome. So there are two pathways. There are the secretory pathway and there is the non-secretory pathway. For the secretory pathway, there has to be a signal, and this signal is right here, the SRP. This is the signal where it will bind to the newly synthesized protein right here. So it will be bound to this part. Now the endoplasmic reticulum would actually recognize that sequence on the protein, and there would be parts here where the where it has some kind of a receptor where it will bind here. So the SRP will bind here, and this signal means that the translation process would actually be occurring on the endoplasmic reticulum, into the rough endoplasmic reticulum. So ribosome will actually come here with the mRNA that it has, and it will keep synthesizing the protein, right? So after that, the protein is already synthesized right here. This is our folded protein, or semi-folded protein, and as you can see here, these black dots, these are the chaperones, and they are on the membrane of the endoplasmic reticulum. These chaperones are so essential for the folding process of the protein. So it gets that newly synthesized protein right here and it tries to fold it. So it goes through cycles of folding the protein, and if there is misfolding every single time after trying to fold it, what's gonna happen is that it will actually put on that protein now there are a few fates that will happen to the protein. So if the protein is misfolded after the trials of the chaperone to just fix it, if it's not gonna be fixed, there will be a signal on the protein to exit the endoplasmic reticulum right away. So it will be exiting the endoplasmic reticulum, and right there, so this is our misfolded protein, okay? And there will be a specific signal on this protein when it will actually go out of the endoplasmic reticulum. It will be ubiquitinated. So it will be polyubiquitinated. So this is our ubiquitin. It's a type of protein that keeps binding as a signal for degradation on the proteins that are misfolded, right? So this is a signal. Well, if there are 2 ubiquitin or 3 or 4, there is no problem. This doesn't mean that it is a signal for degradation. But when it's polyubiquitinated, then this is definitely a signal for degradation of the protein. Ubiquitinated. Okay, so right now, what's gonna happen, this is actually a signal for the proteasome. And what the proteasome does, it actually degrades proteins, only proteins. It's specialized for the proteins. And by the way, proteasome and ribosome are actually 2 organelles. They are classified as organelles despite the fact they are not having membranes. So this is the first fate, which is degradation of protein. Now, what if the protein was successfully folded? What's gonna happen? What's gonna happen next? That there is a post-translational process that the endoplasmic reticulum is responsible for, which is the N-linked glycosylation. And the N-linked glycosylation is actually adding a polysaccharide on the protein. And this process is really essential for signaling this specific protein that it was successfully folded, so that it could actually move on to the next step, which is going to the Golgi complex. So for the Golgi complex, there are two ways. There are actually two ways here. So there is the antrograde. This is the antrograde, which is moving forward, transport, and this is the retrograde. Okay, so for the antrograde, what is responsible for the antrograde is COP2, and this is a transporter. What's responsible for the retrograde going back, COP1. Okay, this is also a transporter. So if we're going antrograde with the protein that we had here, this is the good protein, it's folded, and it will actually enter to the Golgi complex, and there will be another post-translational modification, which is called in this one O-linked glycosylation. Well, the difference between this and the other one, the N-linked. So the N-linked, the reason why it's N, it's actually coming from nitrogen, and the reason why it's O, it's coming from oxygen. So the N, it's actually using the amino acids that contain amide group, which is like asparagine and glutamate. And for the O-linked, it's actually using the amino acid that contains OH. Most likely, it's using serine, and also it uses thyrine. So these are the two amino acids used. So right now, after that step, there is also another fate that could lead to the degradation. Now, if the protein had reached so far, till this far, it will actually it could actually get degraded, and there will be a signal on the protein that you need to be degraded. And in this time, it's not going to go to the proteasome, it will go to the lysosome, where it will be degraded. Now, for the lysosome, also the lysosome has its own acid, right, which is hydrolase. So acid hydrolase, right? The acid hydrolase that is specific for the lysosome. How is actually this enzyme had reached to the lysosome? Well, there is a tag which is called the M6 phosphate. It's mannose 6 phosphate. Acid hydrolase is actually sensitized in the endoplasmic reticulum. So it goes to the Golgi complex, and in the Golgi complex, it actually adds a tag which is called mannose 6 phosphate. Okay, right now, when this specific enzyme or protein gets out of the Golgi complex, the lysosome will actually be able to recognize it because it has the mannose 6 phosphate. Now, there is a disorder if mannose wasn't phosphorylated. What's going to happen? So if mannose wasn't phosphorylated, then lysosomes are not going to be able to use to recognize the acid hydrolase coming from the endoplasmic reticulum, right? So lysosome would actually be useless. It would not degrade the whatever it's needed to be degraded. So there will be accumulation of a lot of components that it's not needed, and most likely it affects the eyes. So it affects the eye lens. So back to the protein in the Golgi complex. So right now, this is one fate, which is the degradation. There is another fate that if the protein had reached that far, it could actually go out of the Golgi complex, and it could actually go to the vesicle. Okay, so it will go to the secretory vesicles, and there it could actually use exocytosis and exit the cell. Now these proteins are like what? These proteins are like insulin, they are like glucagon, they are like whatever enzyme that is actually used, because the enzyme mostly they are proteins, and this is how the protein is actually being exported, or the enzymes are being exported from the cells. So this is the secretory pathway. Why is it called secretory? Because it secretes, like here, it secretes the protein outside of the cell. Now back to the first step. As I mentioned, there is a signal which is called SRP, recognized by endoplasmic reticulum. What if there was no signal and the ribosome started to actually translating the mRNA? Then in this case, it will follow the non-secretory pathway. There is no signal that the endoplasmic reticulum recognized, so it would just go and do the non-secretory pathway, where it will just stay and it will either stay in the cell or the cytoplasm, or it could actually go to the nucleus, go into the nucleus for regulation and gene transcription. So these kind of proteins are actually so useful for the cell function itself. And that's it.